This is DNA or deoxyribonucleic acid. It is a hereditary material in humans and almost all other organisms. Nearly every cell in a person's body has the same DNA. Most DNA is located in the cell nucleus, but a small amount of DNA can also be found in the mitochondria. A DNA is made up of many nucleotides. A single nucleotide is composed of three functional groups: a nitrogenous base, a sugar group, and a phosphate group. Nitrogenous bases are of four kinds: adenine, thymine, guanine, and cytosine. The pentose sugar present in DNA called deoxyribose contains 5 carbon atoms. The third component is phosphate group, which contains phosphorus and oxygen. As already I mentioned, the basic building blocks of DNA are nucleotides, which are composed of a nitrogenous base, a deoxyribose sugar, and a phosphate group. Deoxyribose sugar is linked to the nitrogenous base by glycosidic linkage. The phosphodiester bond joining two successive nucleotides in the same strand of DNA. This bond is formed between the deoxyribose sugar of one nucleotide and the phosphate of the next. When nucleotides join together in a series, they form a structure known as polynucleotide. Let us see how can we construct a DNA molecule with these constituents. To make the DNA strand, I am using jewelry wires and different kinds of beads. First, pick up the bead which represents sugar molecule and put it through the string. Then take the bead which represents phosphate and fix it at the left side of the sugar molecule. Take the third bead, the nitrogenous base, and add it at the right side of the sugar molecule. So we have completed one nucleotide in which a deoxyribose sugar molecule attached to a phosphate group and a nitrogen containing base. Here you can see two nucleotides. In the next step, join phosphate group of next nucleotide with the third carbon of the previous deoxyribose sugar. So I hope you understood that nucleotides are linked to one another in a chain by phosphodiester bonds between the sugar of one nucleotide and the phosphate group of the adjacent nucleotide. Phosphodiester bonds make up the backbones of DNA. A phosphodiester bond is a covalent bond between phosphate and two sugars. One of the strands of DNA has been created here. It is a single linear strand of nucleic acid sequence. They do not have hydrogen bonds and are thus less stiff and also less stable. The phosphate end of the DNA strand is called as 5' end and the deoxyribose end is called 3' end. This strand is in 5' 3' direction. A phosphate group and a deoxyribose sugar make up the backbone of the nucleic acid strand, and attached sugar is one of the nitrogenous bases. If you know the sequence of the bases on one strand of a DNA double helix, it is a simple matter to figure out the sequence of bases on the other strand. Because the nitrogenous bases always pair up in a particular way called complementary base pairing. Thymine pairs with adenine and guanine pairs with cytosine. Two nuclear bases bound to each other by hydrogen bonds then. DNA is always synthesized in the 5' to 3' direction, meaning that nucleotides are added only to the 3' end of the growing strand. So you can notice here the new strand is proceeding in 5', 3' direction. Nucleotides are arranging based on the complementary base pair in template strand, which is in 3', 5' direction. Synthesis of the growing strand involves adding nucleotides one by one in the exact order specified by the original strand. Remember that one of the key features of the Watson-Crick DNA model is that adenine is always paired with thymine and cytosine is always paired with guanine. Here we can observe two strands of DNA run in opposite direction, which means that one strand runs in 5', 3' direction and the other runs in a 3', 5' direction, called anti-parallel. DNA strands are anti-parallel because of the presence of hydrogen bond. Hydrogen bond binds the complementary bases. The main reason why DNA strands are anti-parallel is due to the high level of stability achieved in an anti-parallel configuration. At the end, we have completed the model of double-stranded DNA. It consists of two polynucleated chains whose nitrogenous bases are connected by hydrogen bonds. Each strand mirrors the other as a result of the anti-parallel orientation of the sugar-phosphate backbones as well as the complementary nature of the A, T and CG base pairing.